Coryllium pisci. It is a microcyclic fungus which exists primarily as a mycelium. What is microcyclic fungus means? This need only one host for completing its complete life cycle. So these primarily mycelium is having the uridium and iridospores. These iridospores are oval in the shape and oval to round in the shape. It contains the uninucleate produced singly on iridospores and uridia. These can germinate to form the hyphae which enter. Here you can see the hyphae. These hyphae can enter the leaf through the stomodial tissue, means through the stomata. The hyphae grows intercellularly, name itself indicating intercellular, means inside the cell. It goes inside the cell of the host tissue and extending hastoria into the host cells to absorb the nutrients. With the help of the hastoria, it absorbs the nutrients from the host cell. Iridospores are spread through water droplets and wind. The spreading of this disease is through the water droplets and wind. This disease is commonly during this disease commonly occurs during the winter season. The disease severity is mostly increasing with the age of the leaf. Here you can see the transfer section of the infected leaf, how the infected leaf looks like. As just I told you, the how the iridospores looks like. See here, iridospores contains the thick exine and thin intine that protects the iridospores. And here you can see the germ tube. Once it will fall into the iridospores, the liberated iridospores fall onto the host. It will germinate and reproduce like this. And how the symptoms look like. Here you can see the infected leaf by the leaf rust. The symptoms are a very dangerous leaf disease affecting mostly the mature leaves. The food of late age silkworms. It leads to the loss of 10 to 30 percentage. The pathogen produces numerous here see pin shaped um, pin shaped pinhead size circular to oral brownish to pink black eruptive lesions on the lower surface of the leaf. And in the latter stage, means advanced stage, the leaves becomes like yellow color and fall off prematurely. Why? Because the chlorophyll content is completely utilized by the pathogen. So how to control this one? Timely utilization of leaves without delaying the leaf harvest during the winter season. Wide spacing in plantation is another way to reduce the disease incidence. Spraying of fungicides like Dinocap and carbendazole at 0.2 percentage is also helpful, and the safe period is 7 days. Diseased leaves, shoots are cut and burned. Next, coming to the white rot fungal disease. How white rot fungal disease looks like. Here you can see the mycelium of the white rot disease. This is caused by Roselinea uh, necatrix. This fungus reproduces asexually by forming the chlamydospores, sclerotia, and conidia. Under the favorable conditions, it also reproduces by sexual reproduction by producing asci and ascospore inside the perithecium. Each asci has 8 ascospores. Here you can see the ascospores. This is the reproductive cycle, that is sexual reproduction. And 8 ascospores. The hyphae emerging from the different spores infect the primary roots and later spread to the whole shoot system, causing the white root rot. The sclerotia and the sexual spores remaining as the resting spores in the soil, dried root and stump parts in unfavorable condition. The disease is favorable condition causes the plant, leads to the plant death. And what are the symptoms? Here, the diseased mulberry plants become very weak and stunted growth of the leaf buds and feeble growth and withering of leaves means suddenly cut down of the leaves taken place, weak growth of the plant and the plant dies very soon. The stump region of the affected plants is covered with whitish gray mycelium mat. Here you can see the infected mycelia on root surface and interwound mycelia with conidia. That leads to the death of that particular plant. When coming to the next root rot, violet root rot fungal disease. Here you can see the pictures, clear pictures. Disease cycle of violet root rot. This violet root rot is um, is also a fungal disease. The hyphae are purplish in septate and terminate with fructification bodies called basidia. Each basidia has 4 basidiospores. Here you can see the basidia and basidiospores. And basidiospores at the top. Basidiospores rapidly germinate in wet soils and raindrops, and the hyphae enter the new root system and spread the disease. The germinating spore becomes the nutritive hyphae, which can entrap, which can take up the nutrients from the host and spread the disease. And the hyphae remain for a long time on the dead root tissue and germinate under the suitable condition. This pathogen can also live in many other host plants and perpetuate indefinitely. Symptoms: withering of the leaves and collapse of the plant during the rainy season. The epidermal tissue of the roots is covered with violet colored mycelia mat. As the disease advances, the root spreads to the xylem tissue. How to control this one means uh, mainly root rot disease bring about the rotting of the root takes place. These are dangerous and killer diseases of mulberry. The affected plants suddenly wither and dry. The diseased plants are immediately uprooted. Means that we have to taken from the field, and remaining stumps and root portions are to be collected and burnt. A meter deep pit is dug around the deceased plants to excavate soil and piled away from the field to prevent disease spread. The soil around plant should plugged, leveled and disinfected with chloropicrin. Small ventricle holes 3 to 4 deep are made and chloropicrine solution half kg per 108 square feet we should have to take and put it in the holes are closed with the soil. The field is sprinkled with water. The chloropicrine fumigates the soil and disinfect the disinfection uh, made by mixing the calcium cyanamide with the soil uniformly 75.2 grams per 36 square feet area should be followed. So firstly we have to disinfect with the chloropicrine, then we have to apply the calcium cyanamide into the soil uniformly. The new saplings before planting are treated with 20% of lime water or 1 percentage copper sulfate solution for an hour washed with water. These are the controlling measurements of this disease, that is violet root rot disease. Here you can see the violet root rot disease, that is interwoven mycelia with basidia. Here you can see the root surface with mycelia that is already rotten, and this is the way the basidiospores can get disseminated. These are the basidiospores, and the interwoven mycelia with basidia and basidiospores, the young basidia, and matured basidia with basidiospores. The another disease which is caused by the Fusarium pallidorosium. This Fusarium pallidorosium and Fusarium lateridium and Fusarium mori and Fusarium oxysporium and Fusarium roseum. These are the disease causing agents, that is fungal pathogens which can cause the disease in the mulberry. It can able to produce both the micro and macroconidia. Here you can see the mulberry leaf affected by the twig blight and mulberry stem affected by the stem canker. So what these two diseases? One is stem disease and another one is leaf disease attacked by the fusarium. So as just I said, this is the disease, twig blight disease caused by the Fusarium species. So this can able to produce the microconidia and macroconidia. Microconidia are small, elliptical or curved and unicellular, whereas macroconidia are linear, curved, pointed at both the ends, thin-walled and septate. Chlamydospores are spherical and thick-walled, are formed within the host tissue. All those spores germinate in water to form germ tube. Chlamydospores can survive in adverse climatic conditions for a longer period. This disease can exist throughout the year and cause 5 to 6% of the loss. When comparing with the other diseases, the percentage of loss is bit less. So what are the symptoms? As just we uh, discussed, see here, twig blight. So when the initial stages, leaves shows marginal blackening. In the margin, blackening may be happen with the spores and burning, and at the latter stage, the complete burning and defoliation taken place. The affected twigs become feeble and fragile with irregular black longitudinal patches. Those we can call as lesions on the stem surface. Further, these lesions resulting in splitting and drying of twigs. Birds become rotten. What are the controlling measurements? Fungicides like captafol, mancozeb may be used as a foliar spray about 0.2 percentage and soil drenching with 0.5 percentage. When coming to the mulberry stem affected by the stem canker, here you can see it is almost looking like the cancerous lesions on the stem. So it is commonly caused by Botryodiplodia theobrome. It is a fungal disease. The causative agent, the canker causative agent is Botryodiplodia theobrome. What are the symptoms? Greenish black lesions appear on the stem in the initial stages, which changes to dark black, hard carbonaceous structures. What are the controlling measurements to prevent cuttings from being infected with the stem canker fungus? They are soaked in 10 ppm solution of Bavestin. Bavestin is the best fungicide that we can use in the tissue culture also for 12 hours washed with water before planting. Affected stem should not be used for the preparation of cuttings because they do not sprout. The next one is root knot nematode disease by parasite disease, that is Melodogyne incognita. This is a nematode. It has the three stages in life cycle. One is egg, larvae, and adult. Before going to the further, here, this one, see this one. Uh, this is how the root looks like when it is affected by the nematodes. As just I told you, yes, there are the life cycle stages. One is egg, larvae, and adult. Here you can see the egg, larvae stage. This is the second stage and the adult stage. These are the eggs, larvae, and adult stage. So, generally, this organism enters through the root, the hole made by the stylet, and settle in the subepidermal layer in the second stage. The first stage is egg stage. The second stage is female larvae found in the soil, which root of entry is the hole made by stylet and the settling subepidermal layer. It feeds on parenchymatous tissue. It also stimulates the cells to undergo repeated division, leading to enlargement, thus forming a cancerous knot or gall-like structure on the roots. Female larvae undergo 4 molts to become adults. Each female lays 200 to 322 ellipsoid eggs covered with gelatinous material. Eggs hatch the larvae and are liberated into soil under the favorable conditions. It takes 30 to 40 days to complete the life cycle. It can repeat the life cycle 2 to 3 times in its lifespan. The soil temperature, the favorable soil temperature is 15 to 300 degrees centigrade and moisture 40 to 60 percentage that encourages the parasite growth. The nematode infestation is more in irrigated mulberry farming. So the leaf loss is 10 to 12 percentage. What are the symptoms? Here, as you are seeing in the picture, the more prevalent in the disease is more prevalent in the summer following the rain. The degree of infestation is up to 80 percentage in various states. This parasite causes alteration in plant physiology. The affected plant shows stunted growth, means short growth, and marginal necrosis and yellowing of the leaves taken place. The underground symptoms are formation of characteristics knots or galls on the roots. Small knot indicates single infection and large one multiple infections. The parasite damage xylem and phloem tissues resulting in disruption of water and food conduction. How to control this disease? It can be controlled by deep plugging or digging of infected garden during the summer to expose nematode eggs and larvae to direct sun. Further, mulching, application of green manures, compost, oil cakes are to be used to control the infestation. Application of nematicides like aldicarb or carbofuran at the rate of 30 kg per hectare in the year in 4 equal splits along with fertilizer is the good combination to control the worm. The leaves can be used after 40 to 50 days of application. Soil fumigants like methyl bromide, DD mixture and DBCP are also effective to control soil nematode population. Our next one is mulberry pest, Diacrista oblique, which is caused commonly caused by Lepidopteran pest. Here you can see the Diacrista obliqua larvae, how this caterpillar affects on the mulberry. In the life cycle, it yields 1000 to 1200 eggs that are green in color with a metallic shining color and led by the female moth in small groups on the lower surface of the leaves. These eggs incubate in 5 to 7 days. So the minimum length of the larvae is 0.2 centimeters in length. However, we need the main parts like the pupa is found inside the thin silken cocoon formed by the interwoven shed hair of the larvae. The pupation occurs in soil and lasts for pupation occurs in soil lasts for 1 to 12 to 14 days. The pupa is dark brown in color, measures 2 centimeters long. The emerged moths are light brown in color with wings having scattered black spots. The abdomen is brick red with rows of black dots on lateral and middle dorsal sides. The moths are nocturnal. How to control these moths? Collection of for destruction of eggs masses and gregarious young instar caterpillars. Early stage larvae are identified by dried leaves in the fields. Deep plugging and flood irrigation is useful in killing the pupa with pupae in soil. In biological control, Apantles obliquus is found to parasite the larvae by laying the eggs on the pest body. The parasites puncture the host body to come out from the pupation, thus kills the host. So that we have to spray 0.2 percentage of DDVP or dimethoate on mulberry plant leaves is effective, and farmers are advised to use the leaves after 17 days of the pesticides spray. And there are other organisms which can cause the major loss to our mm, mulberry is gessites. Here you can see the gesside organism here. That is a that is Emposca flavescens, which is belongs to the Hemiptera family, commonly called as leaf hopper. It attacks during the October to May, and it directly suck on the plant sap from the veins on the underside of the leaf. While feeding, a virus is introduced by the hoppers into the lead, which cause hopper burn spots. These spots are triangular in shape with brown at the tip and end of each vein, indicating hopper bite. The affected leaf rolls and becomes scratched. Becomes scratched. These spots slowly spread and complete the leaf. During this stage, the main vein will be green in color. The tender leaves are damaged first, and the latter lower leaves causing with the damage to the crop. Here you can see the life cycle. Adult female lays pale yellow in color, elongated shaped eggs on the lower dorsal side of the leaf below the epidermis, and hatches four eggs in 4 to 9 days. The newly hatched nymph resembles like a adult but wingless. It is in pale green in color. That already looks like a leaf. These nymphs start eating the leaf and grows slowly on the same leaf. It mounts 4 times and becoming adult. The adult is in pale green in color, which measures 2 to 4 micrometer in length with tapering posterior end. The adult and nymphs move sideways. These pests jump to reach other leaves or other plants. The pupation takes place on the leaf itself only. So, what are the controlling measurements? Arrange the light traps to trap the adult gessites, and sprinkler irrigation, spraying of 0.05% DDVP. The safe period is after 3 days of the spray, and dimethoate at 0.1 percentage. The safer period is 10 days of the spray to control the pest. The next one is mealybugs. This insect is Maconelicococcus hirsutus, which belonging to Hemiptera and are the vectors of the virus disease, that is Tukra disease of mulberry, considered to cause by a virus for which male bug is a main vector. The plant affected by Tukra show curling of leaves at the growing point, thickening and shortening of nematode region, and resetting of leaves at the apical end. These organism lays the eggs about 350 to 500 eggs in a loose cottony terminal oviduct during a week's time. The eggs are elongated and red in orange in color depending upon the climatic condition. The eggs hatch in about 5 to 10 days. The newly hatched crawlers are orange in color. The body is covered by white powdery secretions, giving the name mealy bug. The females have three while males have four nymphal instars, which are passed in about 25 to 26 days respectively. The adult reproduce parthenogenetically. They do not feed but mate and die in 2 to 3 days. These attack in summer months. The leaf yield is tremendously reduced and is depleted in nutritive values. The leaves are wrinkled and dark green in color. Here you can see the nymph and adult female and adult male, and how the plant looks like when attacked and affected by the mealy bugs, that is Tukra diseases. Here you can see the spring like structure of the leaf. Then how to control this one? The main the affected leaves and affected shoot are cut and burnt, and also spraying 0.01 percentage of parathion followed. The safe period is 13 days of the spray. Soil application of 4-8 at the rate of 4 kg per hectare after pruning. Safe period is 45 days of the first application. The biological control is carried by introducing predator like Cryptalemus and this is about the melee bugs and tukra disease. The other one is scale insects. Here we have the scale insects. Here you can see the scale insects, the male scale, female scale, and the dorsal view and ventral view of the female adult. And scale insects is attacked by the attack on the mulberry branch, that is E. Here you can see the E figure. So the controlling measurements is, as just we discussed here, cutting and burning of the branches, but there is a different type of controlling measurement for this disease is diesel oil and soap emulsion about 1:3. If you will take 10 ml of diesel oil and take 30 ml of soap emulsion and swabbing on the stem to dislodge the scale insects, because it just um, presented on the stem, that stick onto the stem. So for detaching that one, we need is soap and diesel oil emulsion and lime sulfur. We need we need to swab the stem. So other solution is lime sulfur swabbing on the stem, spraying of 0.05% malathion with a safe period of 10 days, and release of predator, that is Chiloborus cuvanae, and spreading of parasite fungus Atactium indicum is also helpful. And the other one is thrips, that is the Pseudomonas mori. This is the thrip and nymphs. Uh, so how to control these thrips and nymphs? Sprinkling the sprinkler irrigation, spraying of 0.2% of DDVP twice at weekly intervals to kill the nymphal and adult stages. And leaf is used after the 7 days. And other one is mites. Here the mites are like Tetranicus equatorius and Tetranicus talarius. These these are the four pair of legs in adult stage that feeds on the plant, and the presence of my spider mite on the mulberry appears silk. In severe infestation, the leaves lose their green healthy color and appears rusty and finally dry and fall. How to control this one means sprinkling, irrigation, spraying, zolone, theodone, and at 0.05 percentage, the safe period is about 9 days. Next one is stem gilder beetle. Here you can see this is the nocturnal one. That this is the common pest in the India by this beetle. Here you can see how to control this stem gilder beetle. This is affected the affected branches are cut and burned. Swabbing of base of branch with 0.1 percentage of BHC. The safe period is 11 days, or 0.1 percentage of malathion emulsion. Safe period is about 13 days. The other organism or Meloceros species. This gives the major damage to the plants, and here Meloceros species, we have three or four Meloceros species. Those are M. discolor and subfasciatus and viridanus. So the controlling measurements are digging of soil up to um, 7 to 8 centimeters to destroy the eggs, grubs, and pupae, and spraying the phenolphus about 0.01 percentage. The safe period will be 7 days, and 0.02 percentage methyl parathion, and the safe period is 10 days, and 0.02 percentage malathion. The safe period will be 8 days. Next, the common grasshopper, Neorthacris. This is also lay the female lays 6 to 8 egg pods. This is also wingless adult, which looks like green in color. This is also uh, gives the major damage to the mulberry plant. What is the controlling measurements here is exposure of eggs masses by deep plugging and spraying of 0.5 percentage of BHC. The safe period will be 10 days. The last one is nutritional deficiency and control. What are the nutrients and how to control the deficiency? Here, after all these infections, here we are discussing about the nutrient deficiencies and control. What are nutrients? As we already discussed, NPK, nitrogen, phosphorus, and potassium, calcium, sulfur, zinc. All these are the major elements which plays the major role for the growth of, for the healthy growth of the mulberry plants. So what if the any nutrient uh, has the deficiency by the plant? First, coming to the nitrogen, when the plant has the nitrogen deficiency, it looks like retard the development of the roots, reduction in size of the branches, slow the weaker growth, and plant with the less branching and stunted appearance of the plant indicate the deficiency of the soil nitrogen. Young leaves show chlorosis. Stem is slender, yellowish green with stunted root growth. Nitrogen is an important nutrient for plants and accounts about 1 to 4 percentage of the dry weight of the plant. It also forms a biological constituent of all the proteins, enzymes, and chlorophyll, vitamins, and the hormones and nucleic acid. It is essential to increase the content of water in plant tissue and decrease the percentage of calcium in them. The critical level in the soil below which deficiency symptoms occur for the mulberry is 8 kg per acre when the nitrogen is in the form of nitrate and 29.5 kg per acre when it is in the form of ammonia. So the controlling measurements, application of nitrogenous fertilizers like urea, ammonium nitrate, potassium nitrate or calcium nitrate at appropriate dosages is recommended for the correction, means deficiency. When coming to the potassium, what are the symptoms? Stem and root system becomes very weak and becomes like slender. The young leaves become small, shows marginal scorching, and the latter become corrosive, means coarse, non-juicy, necrotic when they mature. The deficiency causes intravenous chlorosis, retards photosynthesis and production of new tissues. The plant may not show good growth even if other materials or elements are available in the soil. This element is highly mobile, do not form any structural component of the plant, but exists in free state as a cation. It activates many enzymes and regulates stomodial closure mechanism, photosynthesis, cell hydration, synthesis of carbohydrates and proteins, and catalyzing nitrate reduction. Its role is important to promote thick outer walls in the epidermal cells and tissues which resist collapse. The leaves show rusty brown patches and are deficient in potash, protein, and sugar contents. The diseased leaves are poisonous to silkworms and cause the disease called Fletchery disease. So, controlling measurements, application of potassium fertilizer is useful in the curing the element deficiency. Here you can see the structure flow chart, identification of the deficiency symptoms. Here, when old release, when uh, nitrogen, phosphorus, potassium, manganese, magnesium and molybdenum, it looks like a dead spot, no dead spots. Here you can see it's a simple flow chart. So next coming to the phosphorus. Here the symptoms of deficiency of phosphorus. The deficient plants show intravenous chlorosis of older leaves. The chlorosis spreads throughout the leaf. This is followed by the marginal necrosis and defoliation. The stem will be slender without fresh growth and the root system becomes like stunted. The decoloration of the stem and the leaves to reddish or purplish color. This element is a component of nucleic acid, protein, and phospholipids and necessary for metabolic activity. It strengthens the stem and prevents the lodging of plant. It influences plant vigor, crop quality, resistant against the diseases. The minimum level of phosphorus required for mulberry growth is 22.6 to 29.5 kg per acre. The controlling measurements, it can be cured by phosphorus fertilizers like triple phosphate, super phosphate. All these are corrected by sufficient supply of NPK, nitrogen, phosphorus, and potassium to the soil at the appropriate time. The next one is calcium. What are the symptoms of calcium deficiency? It causes defoliation of young leaves with necrosis along the veins. The latter stage leads to abscission of leaves. The leaves become pale in color. The plant shows stunted growth with woody stem with yellowish tips. The roots become stubby and dry. It is an important element of all the plant cell membranes and concerned with its permeability. This element controls all division and formation of cell wall by its presence in the middle lamella. It promotes the activities of soil bacteria to form nitrates from organic nitrogen and to fix atmospheric nitrogen. It is also promotes the good root system. It reduces the harmful action of iron and manganese to the plant roots. Plants grow in calcium-rich soil when subjected to moisture stress, lose their water slower than those grow with ordinary soil. Thus, calcium is an efficient element for plant growth. How to control? Liming the fields with the requisite amount of lime corrects the deficiency. Application of calcium nitrate also serves the purpose. The next one is sulfur. It results in the slightly chlorosis of the leaves with subsequent abscission and abnormally long and woody stem roots finally leading to arrest of growth. Sulfur is necessary for the synthesis of some amino acids, coenzymes and vitamins, also formation of chlorophyll. When sulfur is oxidized to sulfates, soil becomes acidic and growth and survival of some bacteria, fungi in the soil are discourages. How to control? Application of gypsum or ammonium sulfate at the appropriate dose is recommended for the correction of, and also factum phos 15 percentage or zinc sulfate 15 percentage can also be used. The other element is zinc. So what are the symptoms if it gets deficiency? Young leaves shows intravenal and yellowish spots. Regularly shaped leaves shows short internodal distance, giving a bushy appearance to the plant are observed. It regulates the activities of the enzymes involved in the formation of IAA, indole acid, the natural growth hormone. So it affects on the formation of natural growth hormone IAA, that is tryptophan. And it is necessarily for synthesis of proteins, nucleic acids and carbohydrates. It also regulates stem elongation and cell enlargement. In addition to the above said, many other traces elements may also necessary for the healthy growth of the mulberry plants. These minerals support good vegetation growth of the quality, quantity of the leaves and building resistance to the diseases and pests. A close and methodical observation, timely supply of required mineral elements, and proper cultural practices prevent the occurrence of the deficiency of the diseases and boost the quality and quantity of the leaf produced by the crop, which can be utilized to feed silkworms to get good quality and quantity of the couples. What are the controlling measurements for the zinc deficiency? Application of the zinc sulfate at appropriate dose for the correction of this element is recommended. Further soluble zinc salt or zinc chelates and zinc sulfates can also be used. So here we have some questions. White root rot disease is caused by Rosalina necatrix and Phylactinia. Here we have the questions. Phylactinia corelia causes powdery mildew disease. And leaf spot disease is caused by Cercospora moricola. And stem canker is caused by Botryoidiplodia theobrome. Mention the micronutrients which are required for the mulberry plant growth. Just now we discussed nitrogen, potassium, phosphorus, calcium, sulfur and zinc. Diacrista oblique is also called as Bihar hairy caterpillar. Tukra disease is caused by Macronelicus hysterus. This is about the complete disease management and how the diseases spread to silkworm. Our next topic is silkworm biology. Here in this topic, we are going to discussing about the silkworm life cycle, systematic position, morphology, life history, physiology of silkworm, and how the digestive system of the silkworm looks like, and how the circulatory system and the respiratory and excretory system, how the reproductive system looks like, and what are the developmental stages of the silkworm. So here we are learning about systematic position, physiology, functional significance of each physiological processes, and also the interdependence of each physiological process on other system. And we are also understanding the development of the silkworm. As unit 1, we are discussing yes, we are discussing the silkworm is a cold-blooded animal and its duration of each life stage, growth factors, feeding schedules are completely depending upon the environmental factors like temperature, humid, rainfall, light and air. Further, the insect racial characteristics are also plays a vital role to overcome all these natural hazards. However, life cycle may be delayed due to the above said factors, and some may die without metamorphosis. The chromosomal studies reveal the Bombax mori evolved from the Bombax mandarina. The domestic silkworm has undergone a variety of genetic mutations like ancestors of the silkworm. How the silkworm life cycle looks like. Before going to that, yes, silkworms go through four stages of the development as do most insects like egg stage, larvae stage, pupa stage, adult stage. The adult is imago stage is the silkworm moth. The larvae is the silkworm caterpillar stage. Since the silkworm grows to the much, they must shed their skin four times while growing. These stages are called instars and silkworms, instars and silkworms, uh, instars and the silkworms only eat fresh mulberry leaves. Silkworms do 85% of their eating during their fifth instar as it reaches about 2 to in length. The larvae has increased its size about 10,000 times more since birth, and its silk glands now make up to 25% of its body weight, ready to spin its silk cocoon. I hope we already discussed about this systematic position of a life cycle. It belongs to the phylum Arthropoda, class Insecta, and subclass uh, Prettigota, and division Endopterygota, and order Lepidoptera, subfamily Bombicoidae, superfamily Bombicoidae, and family Bombicidae, and genus Bombax, species Mori. This is about the systematic position of the silkworm. The characteristic feature of phylum Arthropoda appears in silkworm bombax mori. So, what are the things in this? We'll see. Silkworms are classified on the basis of their native regions and the number of hatchings in as per the year, that is volunteerism, molting, rearing, a rearing period, body markings, and body color of freshly hatched larvae, body color of mature larvae, color of cocoon, color of eggs. But the taxonomic classification is based upon the significant evolution of the organism. It considers the body form, organ system, genetic traits. Silkworms belong to the class Insecta of the phylum Arthropoda. Since phylum Arthropoda, since the insect's body is divided into head, thorax, and abdomen. The body has specific number of segments. The typical insect has 6 segments in head, 3 in thorax, and 11 in abdomen. The head segments are completely fused during the embryo stages to form a head capsule. The other two body divisions possesses movable segments. These insects may or may not have joined jointed appendages and may also possess one or two pairs of wings. Insecta is divided into apterygota and pterygota. The silkworm comes under subclass pterygota. The subclass pterygota is divided into two divisions, namely extopterygota and endopterygota. The silkworm comes under the division endopterygota. There are further divisions formed into different orders. The mulberry silkworm is included in order Lepidoptera by position of two membranous wings with few cross wings, flat scales on the body and the appendages. The larval stage caterpillar is erisiform and with pairs of true legs and five pairs of pseudo legs. The different kinds of silkworms are placed under superfamily Bombicoidae on the following characters. One is maxillary, pulpy, and tymphonal organs are absent, and the wing and locking apparatus or vestigial apparatus are absent, and proboscis are absent, and ketonema is absent. Antenna, especially in males, in the hind wings, SC and R diverge from cell and is connected it to crosswind R1. In forewing, M1 is free. The Bombicoideae consisting of 8 families, of which Bombicoideae includes the economically important insects which produce natural silk of commercial value. Mulberry silkworm comes under the family Bombicidae. The wild silkworm name Bombax mandarina is the ancestor of the mulberry silkworms. Artocarpus incisus is the ancestor of the both Bombax mori and Bombax mandarina. The Bombicidae includes domesticated silkworm along with ancestor studs. Here you can see the morphology. Mm. When coming to the egg, the structure of the silkworm egg, the eggs are tiny and weigh around 2000 eggs to the gram. It measures about 1 to 1.3 millimeter in length and 0.9 to 1.2 millimeter in breadth, and the color and shape and the weight of the eggs, the number of eggs per laying among the different races, also according to the season, larval nutrition of the mother moth, and environmental conditions during the egg layings. Here you can see the anterior pole and the posterior pole and the presence of yolk, periplasm, vitelline membrane, chorion, and anterior polar plasm, nucleus, and micropyle. The eggs laid first are heavier than those laid later by the same moth. With this, today I am going to end this session. Thank you all.